We are on chapter 10, section 4, and all about the regulation of the citrate cycle. So you got to think about the citrate cycle has to be very, very tightly regulated. And so before we start talking about the regulatory enzymes, I want to talk about the mechanisms of regulation, the general mechanisms of regulation for the citrate cycle, right? So you know your end product. Your end product is oxaloacetate. So it's this, since it's the end product and it's the substrate of the first reaction, the flux of the pathway can be controlled by basically resetting the level of available substrate after every turn of the cycle, right? So if you think about the different control mechanisms, right? What can we say? Well, one is substrate availability. ILITY. Okay, substrate availability. And you can put oxaloacetate, right? The other one is you can talk about product inhibition, right? So citrate will inhibit citrate synthase. That's the first step, right? Don't you want a control mechanism where you can stop the cycle at the very beginning so that the cell is not wasting time, wasting resources, making something that it doesn't need? Hey, that's great. So citrate inhibits citrate synthase, right? And then the third one is really what your book has here, which is feedback control of key enzymes. And these are those key enzymes that are regulated, right? So the three main regulatory enzymes are citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. So what's interesting is that all these can be inhibited by ATP. And that's great. That means we have a high energy charge. And so you don't want to go through needless reactions in the citrate cycle in order to realize, oh, I don't really need this anymore. So we're going to inhibit it not only at one place, but we're going to inhibit it throughout the entire pathway in order to make sure that we don't waste our time, right? So the other two that are actually highly regulated as well are pyruvate. So let's see if I can. There's like not enough room on these. Hold on, let me see if I can insert another slide, add page. Okay, maybe, there we go. Okay, the other enzymes that we can actually control are pyruvate dehydrogenase and pyruvate carboxylase. Okay, so these aren't necessarily in the citrate cycle, but they do control substrate availability, right? So we can still use these as control mechanisms. So these enzymes are going to control the amount of our oxaloacetate and our acetyl-CoA, right? And so if those aren't available for the citrate cycle isn't going to go. So you can control what's inside the cycle and you can control things that are outside the cycle. So these are enzymes that we can regulate in order to alter substrate availability. So these technically are classified under substrate availability. Okay, so you just kind of keep those in mind, even though enzymes regulate them. If we regulate these two enzymes, we're regulating substrate availability. All right, so how did I do that? I must have put in a slide in a weird place. Okay. All right, so let's kind of look overall at where we're controlling the citrate cycle. So we know that we're going to have negative and positive effectors of the citrate cycle. You want to be able to promote it and you want to be able to inhibit it. So let's think about negative regulation first, right? If you have high NAD levels compared to NAD plus, right, it's always a ratio. So if we say NADH is going to inhibit it, that means that the ratio of NADH to NAD plus is high. So you have more NADH than NAD plus. This is really signaling that the electron transport chain is not working at full capacity, right? You've got high levels of ATP, you don't need it, so your NADH levels back up, basically. So ATP will also inhibit that. And so what you're going to see is at all these points, NADH, ATP, NADH, ATP, NADH, ATP, right? Those are all the same. We're going to inhibit all different points with basically the same inhibitor. And that kind of makes sense too. And then they have some more specific things like citrate inhibits citrate synthase, right? Acetyl-CoA will inhibit pyruvate dehydrogenase. The product is inhibiting the enzyme. So that's another good feedback mechanism. Succinyl-CoA inhibits alpha-ketoglutarate dehydrogenase. Your substrate is inhibiting your enzyme. Your product is inhibiting your enzyme, not your substrate. Sorry, y'all. Your product is inhibiting your substrate. So whatever you're producing is going to inhibit the enzyme that makes it, right? And so that gives you really nice feedback inhibition. And this is really important because this is not only important for, okay, we need to function, we need to survive and that kind of thing, but we can manipulate the pathways that we understand and we can utilize them to do what we want. So one way, I'll give you one example, is that we have manipulated the citrate cycle in order to use citric acid, right? Citric acid is used in a huge variety of commercial applications, food preservations, reactive acids in polymer chemistry, a stabilizer in pharmaceutical medicines. And so there was a company, Pfizer, y'all know Pfizer, right? A long time ago, figured out that they could use mold, Aspergillus niger, and they can convert. It's this insane, amazing. It's like they start with 140 grams per liter of sucrose, and they get a yield of 115 grams per liter of citrate. That's an amazing amount of product that you're getting, right? That's really, really great. And they have these yeast undergo fermentation. And so we know that when you bring in glucose, you bring in a sugar, sucrose, whatever it is, we're going to go through glycolysis, right? We're going to make pyruvate, and then we're going to go through our citrate cycle. Well, what we can do is we can select for strains of yeast, that of mold, that can't make citrate lyase. So what happens is that these mold cultures are grown in a medium that inhibits part of the citrate cycle. Because it inhibits part of the citrate cycle, what happens is you have a buildup of citrate. So you build up this citrate and you inhibit the rest of this cycle. So this doesn't work. The second half of the citrate cycle doesn't work. You undergo citrate synthase, and then that's kind of it. The reaction conditions, the conditions the mold are in are just inhibited. And then you select for strains that have a high expression rate of this pump that will actually pump citrate out of the mitochondrial matrix, and then it will also export it out of the cell. And so you select for those particular strains, and then these mold are doing your job, right? You're gonna produce all of this citrate, and then we can use this in commercial products. And so we can capitalize on the fact that we understand the cycle, we can inhibit parts of the cycle, and we can make this work for us. So it's kind of a cool application, right? Okay, that is the end of section 4. Y'all have a great day.